Okay, in this chapter, we're going to talk about science and the death penalty. And we're going to talk about fMRI brain imaging. And we're going to talk about DNA evidence as well. So let's start by talking about what we have to prove in court. So to successfully prosecute a case, right, we have to prove that the defendant committed the act, the guilty act, that they did so intentionally, and they did so with a guilty mind. Now, sometimes a defendant will admit to committing the act, but they will argue that the guilty mind requirement doesn't exist. And when they do so, they're using what's called an affirmative defense. That includes justifications like self-defense, consent, and excuses like duress and age. Now, we also have these two concepts of exoneration and mitigation. Exoneration is when someone has been given official legal relief from the burden of responsibility, obligation, or punishment. If later they're deemed innocent, that's engaging that actus reus requirement, so the guilty act. Now, mitigation is the idea that we're lessening someone's responsibility, and usually that means that lessens their punishment. This is engaging that guilty act requirement, the guilty mind. Sorry, not the guilty act requirement, mens rea, the guilty mind. So we see DNA evidence used in terms of guilt and innocence, whereas we see brain imaging used addressing mitigation, that someone is somehow less blameworthy. So arguments around possible innocence were key to some decisions that were made regarding the death penalty in New York, in New Mexico, and in Illinois. Now, judges do seem to be influenced by expert testimony involving biological factors, and it doesn't seem to influence how they feel about responsibility. For example, almost all judges consider psychopathy to be an aggravating factor, but if it's presented with a neuroscientific explanation of that condition, they might use it to mitigate the severity of the sentence. In general, the quote-unquote hypothetical psychopath received a longer sentence than typically given for aggravated assaults to defendants without psychopathy, but judges who were given biological explanations imposed an average of 1 year less than judges who did not get that same information. So judges aren't seeing it as negating any criminal responsibility, but they see it as perhaps lessening the sentence that offender receives. So some scholars believe that it's the hard evidence of science is accepted by the courts. It undermines these ideas of justice because a wholly scientific approach undermines any human responsibility. Are people going to be able to say, my genes made me do it? But keep in mind, there is no such thing as a crime gene. Now, one gene that does come into play here is called the warrior gene. So the MAOA gene comes in 5 versions or repeats, and the 3 and 4 repeats are the most common. Now, this 3 repeat version is a so-called risk gene because it results in low serotonin, and that's the warrior gene. Now, too little serotonin can lead to impulsive behavior, and combine that with high doses of testosterone, that can really facilitate antisocial behavior. Now, Beaver and colleagues found that those with the MAOA-L are more likely to join gangs, and those that did join gangs were more likely to use weapons in a fight. But other studies have found no effect on offending by itself. But when that MAOA-L gene is combined with childhood maltreatment, then we do see very large effects. So the odds of a verifiable conviction for a violent crime were 9 times higher than those with no history of abuse nor the MAOA-L gene. So MAOA-Ls were only 12% of the group's study, but they were responsible for 44% of the criminal convictions among that group. Now, it's important to note that MAOA-L gene is quite common, and it's a risk factor. However, only a small proportion of men join gangs, and fewer carry guns and commit serious criminal behavior. But this is the gene most frequently used as a mitigating factor in murder cases, but it's only been successful once in court. And that offender received a voluntary manslaughter conviction instead of a first-degree murder conviction. So what are genes and how do they make us different? Genes are segments of our DNA that codes for protein, and protein is the stuff that builds, maintains, and replaces tissue in our body. So they facilitate our behavior and feelings by producing tendencies or dispositions to respond in one way or another. So they include us rather than compel us. Now, DNA is 2 strings of nucleotides bonded in that chain that's twisted around each other that we've seen, that double helix ladder. Nucleotides come in 4 types: adenine, thymine, cytosine, and guanine. We get our genes from our parents, who might provide us with both the same or different version of the same gene called an allele. And alleles come in different varieties called polymorphisms, and that refers to differences in the combinations of the alleles located at the same place on a chromosome. And there are minor variations on a common theme. Now, about 90% are single nucleotide polymorphisms, and that means there's a difference in just one nucleotide. We also have a variable number of tandem repeats, and these are tandem repeats of base pairs. So this helps us use DNA fingerprinting, okay? We use polymerase chain reaction, which is a technique that amplifies the copies of a DNA sample so that we have enough for testing. After that DNA sample is isolated in a test tube, 2 strands of nucleic acid are synthesized in the lab, and they're called primers. And they serve as the starting point for replicating the daughter strands. That sample is heated, and it pulls the 2 strands apart, and that's called denaturation. DNA is then cooled, and primers containing all the ATCG bases that are complementary to the target sequence from the sample are added. Then it's brought to another temperature where a special enzyme called DNA polymerase is added to that, and it synthesizes the DNA strands. Each strand then sets about rebuilding the complementary strand to itself as a free nucleotides attached to themselves, and this results in a new double-stranded daughter molecule of DNA. After we have enough copies, they're going to separate these using electrophoresis, and this is the migration of the DNA molecules that have been placed in a gel box with an electric power source. Now, we have to be very careful in the lab because even the slightest contamination can lead to a false positive or a false negative. Results can also be misinterpreted. Different labs may make different decisions. So when an allele drops out, it could be because the DNA is contaminated, it could be because there isn't a match, or there could be some acceptable or unacceptable margin of error. This can lead to a factually innocent person being convicted or a factually guilty person walking free. Now, brain imaging is looking at communication in the brain, and that takes place via tiny electrical cells that run down the axon, which is an extension of the neuron. Between each communicating neuron is a tiny gap called a synapse, and messages are going to jump across that gap, and when they do so, they're translated from electrical to chemical energy. Chemical messengers called neurotransmitters jump the gaps between the sending and receiving neuron. Brain imaging basically turns that into pictures of what parts of the brain are activated. So what happens is people will use an fMRI, a functional magnetic resonance imaging. This shows the structure of the brain and informs us if certain part of the brains contain tumors, are damaged or atrophied, or have some other structural impairment. But it also allows us to see how the brain functions in real time. So we would have you lay down in an fMRI machine and perform a series of relevant mental tasks. You might be given a series of pictures or scenarios, and then you may be asked to respond to questions by pressing buttons on a response box. The fMRI then picks up and stores information about the regions of the brain that are being used. Now, the harder the body works, the more oxygen it requires. So when a particular region is activated, more blood containing oxygen is routed to that region. So the fMRI uses the fact that we see more blood and therefore more oxygen going to that area. And it uses blood oxygen level dependent contrast, or bold contrast. Now, the most far-reaching use of brain imaging to date involves challenging the death penalty for juveniles. Now, we know that most teens who do offend desist as they reach into adulthood. And the New York Academy of Sciences had a conference on adolescent brain development in 2003. And they noted 3 key points. 1, much of behavior characterizing adolescence is rooted in the changes that are happening biologically along with our environment. 2, the lack of synchrony between this physically mature body and a still maturing nervous system may explain this behavior. And 3, teens' sensitivities to rewards appear to be different than adults, prompting them to seek higher levels of novelty and stimulation to achieve the same feeling of pleasure. So puberty is this mark of our transition from childhood to adulthood. We have a lot of physiological and neurological changes. There's a 10 to 20-fold increase in testosterone in males, and we know that testosterone is linked to aggression and dominance-seeking behavior. The ratio of excitatory to inhibitory neurotransmitters becomes unbalanced because the brain is slowly undergoing this process of refining our circuitry to its adult form. So that prefrontal cortex, the front part of our brain that we know is responsible for making judgments and planning, is getting its final coat of myelin. That is a fatty substance that insulates various brain structures that are important for the speedy electrochemical transmission of information. So that's where our moral conscience and our logical reasoning is happening. So as we've discussed before, the juvenile brain is literally not fully formed. So we know that the brain reaches its adult stage in our mid-20s. And then we start to see more adult-like personality traits emerge. People tend to be more conscientious and agreeable, less irritable and neurotic. Neuroscience has shown that a lower level of culpability is attached to juvenile offenders. And this was a major factor in Roper v. Simmons. There were a lot of amicus briefs submitted, in particular from the American Medical Association and the American Psychological Association, and they both offered extensive information on brain imaging and how it shows that the juvenile brain is not fully developed. Now, Supreme Court decisions have to be based on constitutional grounds, and anything that is a non-legal statement is considered dicta, so it's legally not binding. So, the reason for the decision is based on Atkins, this evolving standards of decency. Now, in Graham v. Florida, right, where they're dealing with license without parole for juveniles, they do explicitly refer to the neuroscience as a compelling factor in its decision. Now, what are some problems we have to think about? We tend to think that science is telling the truth, but DNA can, quote unquote, lie. So, as I mentioned before, we have to handle it meticulously, and we have to make sure that we know what we're doing when we're interpreting those PCR results. If DNA provides certainty, then pro-death penalty advocates can argue that major obstacle of what if someone is innocent is no longer going to exist. But we know that a number of police labs have been accused of and found guilty of falsifying and fabricating DNA. We just had a major case with this in Massachusetts in regards to drug cases where literally tens of thousands of cases have now been thrown out. Some labs are incompetent in using PCR, some of them are not interpreting results correctly, and some have cross-contaminated and mislabeled sample DNA. But even without any bias, experts can come to different conclusions. So Dror and Hampkin in 2011 did a study where they submitted mixed DNA to 17 DNA analysts. Only one person reached the same conclusion as the original analyst in the criminal case, that the defendant could not be excluded. 12 concluded to exclude the defendant and 4 were inconclusive. So even if experts disagree in their interpretation, we then have to realize that DNA is not this holy grail of guilt and innocence. Now, what might be some problems with fMRI? The data from fMRI are aggregated. It's information from a whole series of fMRIs done on a series of people. Okay, we can't take group information and assume it applies specifically to an individual. So jumping from group data to specific individuals is an ecological fallacy. It's a mistaken logic that we make when we make an inference about individuals deduced from aggregate data. Neuroimaging evidence relating to which parts of the brain are activated under which conditions and which parts are responsible for which behavior, however, is consistent and strong. But we also want to remember that this is not a direct measure, it's an indirect measure. We also want to remember that correlation is not causation. So just because two things are related or occur together doesn't mean one causes the other. So it is a big jump from talking about impaired judgment inferred from mistakes the subject make on tasks performed during fMRI to a brutal murder performed in the real world. And just because a crime is committed impulsively doesn't mean it's still not committed consciously and intentionally. We also know that this brain imaging often takes place years after the crime was committed. So we don't know if this is the state of that offender's brain when they committed that crime.